Okay, in the interest of time, I think I should get started, and more people may come in while I'm giving a brief introduction. My name is Sarah Partan, and I use she, her pronouns, and I'm a professor of animal behavior at Hampshire College. And I wanted to start with a land acknowledgement, to begin by acknowledging that Hampshire College stands on Nonotuck land. And we'd also like to acknowledge our neighboring indigenous nations, the Nipmuc and Wampanoag to the east, the Mohegan and Pequot to the south, the Mohican to the west, and the Pocumtuck and Abenaki to the north, and many others. And we also acknowledge that acknowledgements are not enough, but we all need to work to do more. So I want to explain the format of the session, which is that each student will have 10 minutes for their presentation and 5 minutes for the question and answer. And audience, please put your questions in the chat. You can just put them up as you think of them and go ahead and populate the chat. This session is situated in the Environments and Change LC, the Learning Collaborative. So we have, this year we're doing learning collaboratives that are our new academic program across the campus with faculty, staff, and students all participating in this deeply transdisciplinary work. So the Environments and Change LC asks questions such as how we repair our environment and reverse climate change, how we care for each other and the planet, how we define a sense of home or embody our place in the world, how we adapt, how we define and measure environmental change, and many more questions, which you'll hear about today from our wonderful Div 3 students, who we couldn't be more proud of for pulling together their Div 3s in such a difficult year and really doing a stellar job. So with that, I'd like to get started and introduce our first speaker, who will be Hannah Ballantyne. So Hannah, would you like to share your screen?
So my name is Hannah, and my Division 3 is called Health Choices Waldorf Families Make During a Global Pandemic and Implications for the Future. And my committee was Dr. Pamela Stone and Dr. Alan Goodman. So an introduction. My position here is that I'm an alumna of the Hartsbrook School in Hadley, Massachusetts, and I take a pro-vaccine stance throughout this presentation and throughout my Division 3 end project. So my question was, how is the COVID-19 pandemic affecting the views and practices of vaccine hesitant communities? And this question began at the beginning of the COVID-19 pandemic, and I wanted to learn about what this approach would look like at the Hartsbrook School last year, and so studied it throughout the course of this year. A little bit of a background on Waldorf education. It is an alternative form of education in private schools all over the world, and it was developed by Rudolf Steiner in 1919. And what I've learned over the course of my Division 3 is that there's a lot to be very critical of in Waldorf schools and in regards to Rudolf Steiner and his racist history and the history of oppression and things that Rudolf Steiner did that are not admirable today. But it's also a complicated issue because the Waldorf schools can be very, very good today. So it's been complicated to learn about and to digest. Here are some photos that are examples of Waldorf classrooms. As you can see, the photo on the left is a kindergarten. It's very pink, it's very beautiful, and there's even a whole science behind what makes a Waldorf classroom, all the way down just to no right angles or rigid angles in the classroom. And that's been very fascinating too to learn about. An overview of my div. So I started off by writing a background for future readers about the COVID-19 pandemic, which was very fascinating to write about, sort of defining what is social distancing and how does COVID affect your body. All of these things were really interesting to write about. I wrote a chapter exploring vaccine mandates, and this was quite interesting for me to dive into. And then the Hartsbrook School as a study site, an ethnography of Waldorf families in response to a pandemic. I did an analysis on that ethnography, and then I wrote a discussion. So right now, I'm going to read the full introduction to my div. The anti-vaccination movement has always fascinated me. While my interest in human health has always been a part of my academics, the early days of the coronavirus pandemic sparked my fascination in this topic. I was fueled by questions surrounding a future COVID-19 vaccine and what may be barriers to inoculating a large percentage of the US population. I quickly came to realize that conspiracy theories driven by anti-vaccine, also known as anti-vax, anti-science, anti-mask, and pandemic denier groups, among others, may be some of those barriers. While determining what may be the biggest barriers to inoculating a large bulk of the public against COVID-19 are out of the scope of my Division III thesis, I hope to draw on examples of past mass vaccination campaigns to contextualize how this one might roll out in the present moment. I have chosen to focus my research on how vaccine mandates affect families and to consider how the blanket arguments that many vaccine hesitant groups are centered on ideas of health freedom, which is also anti-mandatory vaccination. My goals for my Division III are multiple. I am interested in understanding the history and development of vaccines, the mandates that followed them, how effective they were, and how the public reacted to them. Today, with a new vaccine, it is also useful to understand the social aspects of how the public accepts the COVID-19 vaccine. As the development of the COVID-19 vaccine has been a continual process throughout my Division III year, following the trends of what my family, friends, and the media are saying has been extremely informative. This led me to developing my main question: how is the COVID-19 pandemic affecting the views and practices of vaccine hesitant communities? To unpack my question, I explore the health concerns and practices, particularly around vaccinations within the community associated with the Hartsbrook Waldorf School in Hadley, Massachusetts. The Hartsbrook School has one of the highest non-medical vaccine exemption rates in the state, at 25% of the school either being partially or completely unvaccinated. Within my work, I explain the significance of vaccine hesitancy in the Waldorf school communities globally, as well as exploring vaccine hesitancy from other angles, like the far right and far left. I begin my Division III by writing about vaccine mandates to give context for the legal, social, and political implications for vaccines on a broader scale. This encompasses both the science and public health lens of vaccines, as well as the social context in which information on vaccines circulates. I then offer a short ethnography that centers the voices of parents from the Hartsbrook School to give voice to the ways people with privilege and choice to not vaccinate are making decisions and navigating this pandemic. The greatest takeaways for me have been the inherent complexity of the vaccine debate. Vaccines are a Western biomedical intervention that is necessary to protect the common good and requires as many people to take part in it as possible. My Division III gives context for the complexity of vaccines, vaccine mandates, and vaccination in the time of a global pandemic. It also explores my own relationship to this complex topic, having graduated from the Hartsbrook School myself. And finally, as this Division III has been written as the COVID-19 pandemic has raged on, and new, safe, and effective vaccines have been produced at record speed. This project gives a glimpse into the historical archives for one of the Anthropocene's worst plagues. So to finish up the themes from my study, I can sort of give a little bit of a preview for people who do want to read the full study. The four main themes from this were the Hartsbrook School, Waldorf education, and the geographic location of the school. What I found from this was that a lot of the circulation of information really has to do with place and location. Parents would, in my study, there were 6 participants. They described learning about things like homeopathy or other alternative remedies through what they describe as play yard chatter and just discussing these things with other parents at pickup, for example. The second theme was white privilege and socioeconomic privilege. What I found was that a lot of parents discussed their own privilege and were able to recognize that, especially during a pandemic, and being able to send their children to this outdoor school. You can see here it says they have an outdoor classroom option this year, and to be able to pay high tuition to send their kids to this school. The third theme was choice surrounding healthcare and vaccines. What does it mean to have a choice? What does it mean to give your children a choice, like to be vegan with the rest of the family or not? And the fourth theme was science and education. I was really interested in what people described as research and what doing your own research means. Some parents would read journal articles with peer-reviewed research, and some would read Facebook links or listen to sort of anecdotal stuff. And so I was able to really analyze all four of these themes to answer some of my questions about how Hartsbrook's approaching the pandemic. And lastly, I would like to thank the Hartsbrook School and to my committee and my family members and everybody else who's supported me through this year. So now for some questions and answers.
Great, thank you, Hannah. That was wonderful. So please, the audience can put your questions in the chat and we'll see them there. One of the audience members is saying, great topical and local subject, so close to heart and home. Really interesting work. While people are thinking of their questions, I want to ask you what are your, are you planning to take this study further, or what are your next thoughts about what you're thinking of doing?
Yeah, I would love to know if these parents got vaccinated now, because the study was finished in December when, and one of the main questions was, will you, oh, you did this change your opinion on vaccines? That's a good question. The study was performed in December, so many of the parents said, like, yes, I'll take the COVID vaccine as soon as I can get it. Some were still hesitant or skeptical. Some said they wanted to keep waiting longer. So I would want to do, like, a year from now, a follow-up would be fascinating. Mm-hmm. So to the question, did this study change your opinion on vaccines? I was afraid that my opinion would be changed because I read a lot of the side of the anti-vax side, and it was pretty manipulative at times because they'll cherry pick things from past conspiracies that have been proven to be true. Like, they'll compare vaccines to the opioid crisis, and they'll talk about big pharma and various views on health, and it can be very confusing. And so I can see how research really has shown that it takes 5 to 10 minutes on an anti-vax site to be really persuaded and manipulated. So I was afraid my mind would be changed, but I think I only got more grounded in the science and in the evidence.
Mm-hmm. I also see that we have the Q&A open, and some people put questions in the Q&A. So Clay's asked, why are people hesitant? And that is similar to the Matthew's question, who just put in, what were the main factors people didn't want the vaccine? What were the deciding factors? Yeah,
the main factors in my study were parents would say it's too new, it's too rushed, we don't know enough. And so I'm gonna stop sharing my screen. Yeah, they would say it's too new, too rushed, we don't know enough. And now I think it's just become more apparent that there's a mountain of evidence that we really do know enough now. And we do have decades of research on mRNA technology and things like that.
Mm-hmm. And Cindy Gill asks if people had concern about the science, what was the biggest concern about the science that people had?
They just wanted to just keep waiting and see how it affected people. Mm-hmm. Yeah.
And I see I missed Jean's question, but it was similar to the other question that you had answered already about what you conclude about families' reasons for not vaccinating. So,
yeah.
How do you think you could change minds on this issue?
Yeah. A lot of it is through culturally competent and relevant discussions and working with community-based leaders to meet people where they are at and to discuss very valid fears and concerns, especially with people in the BIPOC community and people who have experienced medical racism, to discuss that these are very real concerns that need to be addressed. And so working at local levels with people, community, faith-based organizations.
Yeah. And here's a question.
Steiner School, yeah.
Yeah. Do you want to read it, or should I?
Yeah, healthcare workers. Yeah, maybe Sarah, you pick some.
So in case anybody's listening by phone and can't see the questions in the chat, there was one that said many healthcare workers were hesitant too, which was interesting. And another one saying that there have been California Steiner schools where parents opted not to give their children the measles vax, and that spread the disease. Yeah. Did people discuss that with you?
Yes. So a few parents said that a common question they get as parents sending their children to Waldorf schools is they'll get asked, are your children vaccinated? Because there's been a lot of headlines about under-vaccinated Waldorf schools, particularly especially in California. And so before I even jumped into my study, I read a lot of E.J. Sobo's work, and she's an anthropologist who did a few different studies at Waldorf schools in California, and particularly anthroposophy, the philosophy behind Waldorf schools and that Steiner created. There's some vaccine hesitancy there, as well as just community circulation of information.
We should wrap up to turn it to the next one, but maybe you can answer the rest of the questions in the chat, Hannah. And thank you so much again. That was wonderful. Congratulations. Okay, so our second presenter is going to be Flynn Hibbs. I had the pleasure of working with Flynn on their committee. So Flynn, please take it away.
Great, thank you. One moment. Okay, you can see my slides?
Yes.
Great. All right, thank you. I'm Flynn, and the title of my Division 3 was Ecological Effects of Utility Scale Solar Arrays on Macrofauna in New England. So just to give you a little rundown of what I'm going to talk to you about today, first I'm going to share how I got introduced to researching wildlife and solar, and then I'll talk a little bit about what we know about the ecological effects of solar are, and then I'll move into explaining my Division 3, my question, methods, results, and then I'll discuss my takeaways from my findings. So I started my Division 3 in a way my first year. So when I arrived at Hampshire in 2017, it was the first year that our college's arrays were up and running and powering our entire campus. And it became a space for research and inquiry among faculty and students. And I worked with Dr. Sita Sisla, who was here, and she was investigating the ecological impacts of solar, and I was helping her do field work. And we were visiting the sites often and started noticing tons of animal signs. And I was really intrigued by this. And I asked Sita if we could set up a trail camera at one of the massive fox dens we found inside one of the Hampshire arrays. And the picture you're seeing right now is one of the first pictures I ever got of the fox at her den. And I was immediately hooked. And to explain trail cameras really quick, they're an amazing tool. They're remote sensing cameras. So when an animal passes by, the cameras are triggered by heat and motion. So when an animal passes by, it automatically records a picture of that animal, as well as some other information, such as time of day, temperature, even the phase of the moon. So it gives us a lot of information. And they're really an amazing tool that gives us a glimpse at the wildlife activity that happens all around us that we never really get to see with our own eyes. So, after doing this tiny little pilot work, I was really interested in it, and I spent the next year designing my study, applying for grant funding through Hampshire, and I got more cameras and I set them up at the 2 arrays at Hampshire, inside and outside of them. And with more cameras and better cameras, I was really shocked when I started going through the footage, because I was finding and I was seeing lots of animals and, lots of different species, not only mammals, but birds as well. And it seemed like I was seeing just as many species inside as I was outside. So in my Division 2, I continued this study, and when it came around to Division 3, I knew this is something I wanted to continue focusing on. So that's a little background of how I got into the research, and now I'm going to switch gears and give you just a little bit of background on why I'm even researching this in the first place and why it's important. So the study of ecological impacts of solar is a relatively new one, and there's very little empirical research on how wildlife populations are impacted by solar and utility scale solar, so big arrays that are powering campuses or large areas. So one effect we know that's pretty obvious is habitat loss and fragmentation. In these pictures here, you can see just how large these arrays can be. And so this means direct resource loss for species as well as fragmentation. So populations can be isolated from one another. It can be hard to move across the landscape. So that's one issue. Additionally, the arrays can become areas for non-native species to colonize. So when developers come in and they clear the land under arrays, they don't always reseed with native species. So that can have a detrimental ecological impact. They can also act as ecological traps. So these arrays emit a polarized light that attracts insects and birds that think it's an aquatic water body. And so this can mean that birds come down to land on the arrays thinking it's water, and then there's collisions and fatalities. It can also mean that the insects think it's a water body that they can lay eggs in. And so then when they lay those eggs in an array, those eggs don't hatch, and there can be massive population declines because of that. So there's some effects. What we know about wildlife and solar, the studies that are out present some conflicting findings. So we don't have a clear picture of what's going on yet. We know that some studies have looked at bird populations and have found that they do cause direct mortality as well as decreased species richness and abundance inside arrays. And conversely, another study conducted at one of the largest arrays in the US, they actually found high biodiversity within their array and recorded a vulnerable and endangered species, the kit fox, which you're seeing right now. So we know a little bit, but it's not a consistent effect of arrays on wildlife populations that we see so far. And also, the majority of these studies focus on desert species because that's where planting and installations are concentrated. So we don't know much about how wildlife populations are affected in temperate or tropical zones. So that's where my research comes in to fill this gap. So my research question was, will we see a difference in diversity and abundance of mammal and bird species outside utility scale solar arrays compared to inside? And we expected to see greater diversity and abundance outside the arrays because it's not an ideal habitat, and because of the fencing that is a barrier for species that doesn't allow them to enter the site. So in terms of my methods, I had a total of 24 cameras at 4 utility scale fixed panel photovoltaic solar arrays. And there was always an even number of cameras inside to outside. As you can see in these photos, I mounted the cameras inside the arrays to the steel beams supporting the panels. And then the cameras outside the arrays were mounted to the fencing or to a nearby tree. And the data I'm going to show you was captured from September 2020 to February 2021. Just to give you a little geographical context. So these are where my sites were located. I had one site in Brattleboro, Vermont, and this star in the middle here is the Hampshire College arrays. And I also had another site in Holyoke, Massachusetts, near Mount Tom. So these are the 2 Hampshire College arrays. They were sited on former agricultural land, and they have fencing that does not include a wildlife clearance. So we didn't put in any space for wildlife to move through our arrays. The fencing goes all the way to the ground. The site 2 here on the left is the site in Holyoke. And this site is the only site of mine that had a wildlife clearance. So there was a 6 to 8 inches of space from the ground to the fencing that allowed for wildlife to move through the space. And site 3 is the one that was in Vermont, and it did not have a wildlife clearance. And all of the habitat within these arrays was a grassland habitat. So results, what did I find? So we recorded signs of wildlife creating ways into the arrays by digging under, as you can see in this photo, or by digging tunnels that led them inside and outside the arrays, which was really clever. In terms of abundance, what you're seeing right now is my 4 sites here on the x-axis and the number of individuals we saw. So mammals and birds combined. And the different colors are showing the light green is inside and the dark green is outside. So we saw pretty similar abundance across all sites. And then when we look at species richness, so the absolute number of species we saw, we also see pretty similar species richness inside and outside these sites, with the exception of site 3. And then when we look at diversity, which is slightly different than species richness because we're taking into account both the number of species we saw and how many we saw, so the abundance. And we see that 2 of the 4 arrays have statistically significant results. So site 2, we saw greater diversity of mammals outside the array. And in site 3, we saw greater diversity of mammals inside the array. And then at the 2 Hampshire sites, which are 1A and 1B here, we don't see a significant difference. So we see similar diversity inside and outside. So pretty conflicting results. And then when we look at birds, we see that at one of the Hampshire sites, so 1A and the Holyoke site, we see greater bird diversity outside the arrays compared to inside. But for 2 of the arrays, we don't see a statistical difference. They're pretty comparable in terms of the diversity we see inside and outside for birds. Another study had comparable results. They also found higher bird diversity outside. So that may be a consistent finding in the literature as we
About a minute left.
Okay. We also found important biodiversity with conservation concerns inside, including the American inside arrays, including the American woodcock and the common grackle. So takeaways, what did we find? What does this all mean? So firstly, fencing is not deterring wildlife from entering the space. We observed animals creating ways into the array, and we also found high species richness inside all sites despite the differences in fencing. And there were also signs of mammals and birds nesting inside the arrays. Here's a picture of a bird's nest inside an array. Important biodiversity with declining populations were seen inside an array, which could imply that these arrays have the ability to be a space for vulnerable species to gain resources from. But having said that, we also found that the majority of species we captured were generalist or invasive. So these conflicting results really highlight a need to study community composition before we put in solar and after to get a better sense of how biodiversity and abundance of species changes or doesn't change after we put in solar. Utility scale solar arrays, at least in New England, are not industrial energy wastelands that are totally devoid of all life, and they may actually be supporting many species. Our findings suggest that the habitat within arrays attracts species despite the fencing that could deter them from entering the space and provides resources to some species, mostly generalists. That concludes my talk. I want to briefly thank my committee, Dr. Sarah Partan and Dr. Blair McLaughlin, as well as Dr. Sita Sisla and my collaborators, and all of the people at my solar sites that helped me conduct my research. And I'm really grateful to get to speak with you all today, share my research.
Great, thank you, Flynn. Okay, questions. Please put your questions in the chat or the Q&A. Fascinating research, says Zepha, so important in the great shift to solar. Yes. While people are thinking of their questions, do you want to tell us what you're going to do with this next, or where you're headed next?
Yeah, so I'm hoping to publish this research, and I'm working on a manuscript for that. And next I'll be heading to, in a couple weeks, to Florida to start a research internship down there doing applied ecology work. Spencer says, what's the coolest animal you observed? This past fall, I captured my white whale, which was a black bear at the site in Holyoke. So that was pretty cool. I also captured, what else did I capture? What was the most surprising or unexpected animal you saw? Well, when I was first doing this, I really didn't think that larger predators could enter the sites. And I saw in my first year or two of studying this at the Hampshire sites, I saw eastern coyote inside, and I was so shocked by that. And one time I actually entered the array and a coyote was in there with me. And so it was kind of crazy to think that these large predators can not only find a way inside, but also want to enter the space. And that means there's resources in there that they want to capitalize on. There was a concern about animals disrupting arrays. Do you think this happens? That's a great question. We don't know that yet. And that's a big question in this field is, okay, going forward, do we want to design arrays that allow wildlife to enter the space, to move through the landscape, to access the resources inside? Or do we want to, if it has a negative effect on the animals or on the solar industry, do we want to prevent animals from entering the site? In terms of animals disrupting the arrays, I haven't seen anything like that. And I see woodchucks burrow right on the electrical equipment in the arrays, and they still work. So it could be that they're not disrupting the arrays at all. But that's something that needs more research for sure. Tatiana says, I'm curious. Hi, by the way, hi, Tatiana. Tatiana helped me get into my Vermont site, and I'm so grateful for all of her help. I'm curious as to more specifics about the resources that might be available to some species inside arrays. At the site where you have more birds inside than outside, do you have any cases for why that was? Yeah. So resources that are available is the vegetation, obviously, habitat for, we saw lots of nests and burrows inside. That was a pretty common thing to see inside all of my sites. For predators, the resource is the small prey that's going inside the arrays and eating the vegetation and burrowing in there. In terms of the birds, what we think is happening is a lot of the bird species around here are passerines, so they're perching birds. So the arrays provide a perching habitat. And when we record right outside the arrays like I did to compare biodiversity and abundance, there isn't that perching habitat outside. So it could just be because of my sampling methods, we saw more birds inside, but really maybe there isn't. Could also be that a lot of these really large flocking generalist species were seen inside arrays consistently. And so that may be why we see more abundance of birds inside, but less diversity inside, is because we have these main couple generalist species like your European starlings, your red-winged blackbirds that are entering the sites. I think that answers your question.
Yeah, and I think we're about out of time for questions, but there are a few more. Thank you to everyone who was asking questions. There are a few more in the chat. If you could type the answers into the chat, that would be awesome, Flynn. So thank you very much. That was great.
Thank you.
And we are now moving to Oscar's presentation. So this is Oscar Loge. And Oscar,
hello.
Yeah, great.
Yes, hello. So I will start with a song about earthships. That's that earthship, 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 yeah, that's the earthship, 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 yeah, that's that earthship. All right, welcome. I made that at the Earthship Academy. Can everybody see the presentation? Okay. Where did Zoom go? I gotta share screen. There we are. So, while at Hampshire, my heart guides me to the woods. And in moments of deep peace, I look around and ask myself, where will my food come from? Where will I find clean water? Will I have to chase after a bunny or boil some water? How will I keep warm throughout the night? And basically, how can I survive in a way that does not harm the planet? over the summer, I got to attend the EARTHSHIP Biotecture Academy session in Taos, New Mexico. It was a very cool experience. We got to build these EARTHSHIPs, the self-sufficient housing, and learn about the history of this type of construction, and read floor plans, as well as their projects abroad, building structures that can withstand the worst of climate change, like powerful hurricanes and earthquakes and stuff the earth is throwing our way more and more these days. Yeah. So while there, I got the chance to build community. I made a lot of wonderful friends and repurpose waste, which is a big issue. Make use of things that would otherwise pollute environments, and get some good exercise while what felt like saving the planet by building tire walls, as you can see, and making eco bricks out of non-recyclable plastics. So currently, the built environment does not take into account the movement of the sun. Oftentimes it does not. And when it does, it can be used to heat the space. Heating and cooling systems are often noisy, and our way of acquiring materials, like clear cutting forests, it doesn't show respect for the planet or foster a sustainable way to live. And most importantly, spaces are disconnected from nature. One of my favorite features of EARTHSHIP houses is the greenhouse. They also serve as greywater planters to filter water. And you can grow quite a bit of food in there, like figs, bananas, like rosemary, mint, all sorts of things. I've spoken with some friends at Hampshire who experience seasonal depression. This area can get very cold, so having a space to go where nature is alive inside year-round is an excellent way to keep spirits high and bring people together. Yeah, so the greenhouses are beautiful. I would love, when I was at EARTHSHIPs in Taos, I would love to go in there and scavenge for the fruits and eat them. And it's wild because the outside environment is so dry and barren, and yet indoors there's such vibrant ecosystems. So how does a space like this stay warm all year without requiring fossil fuels? And in the winter, when the sun is low, the sunlight passes in and heats the thermal mass in the space. So the back wall and the floor, and the rigid perimeter insulation, the pink lines, encase the space sort of like a thermos bottle so that the heat does not escape. At night, once the interior space cools and the sun sets, this warmth transfers back into the living environments to provide like natural heating. It's very agreeable to be inside an EARTHSHIP. The exterior of these structures are bermed. They're surrounded by like mound of dirt, and grasses can grow over time. So the spaces are like able to blend into nature. Also, with extreme weather, hurricanes can pass over because the winds are able to go over like a speed bump rather than just encounter a flat wall and rip the roof right off. So it's very important in areas prone to hurricanes. And yeah. In summer months, when it gets when it gets really hot, there is a way to cool the space using like ancient cave technologies. Buried 30-foot long buried cooling tubes draw in outside air, and the buried soil in the berm remains cool, and that coolness is transferred to the air that passes through the tubes. So when it comes into the space, the fresh air drawn in, there's a draft created by opening vents in the greenhouse. And so a natural circulation allows the building to breathe. And when I was in there and in these earthships, you can put your hands up to the vent and you feel this cool air coming through. It's free and environmentally friendly and very, very cool and efficient. Every earthship is equipped with a rainwater harvesting system. There are silt catches to remove debris before the water enters the system. This is important so you don't have to clean them too often and they don't get clogged. Once inside, the water passes through a pretty elaborate filtration system that removes particles, debris, and bacteria that can be harmful to human health. Where states allow this system can clean the water to be potable so you can drink it. It's clean enough to drink. Some states are very picky and about not allowing this. But the water can still be captured and used to water plants. So in Texas in February, a very powerful winter storm came to the area. They were not used to experiencing such cold temperatures, and so their infrastructure basically collapsed, leaving millions without clean water, power, and the homes were incredibly cold because they require heating in often cases to be hospitable. And so this really shows the importance of having buried water systems so that your pipes don't freeze and you're not without water, and having off-grid so you have access, you're in charge of your own survival, you're not dependent on outside systems.
You got about 1 minute, Oscar.
Oh geez. This similar thing happened at Hampshire, where we lost power in the area, heavy wet snow down trees, off-grid solar on earthships. There's a bathroom. No matter what happens to the region, the area will have running water inside and a toilet. GREYWATER. Sewage, which fertilizes planter cells. So my hope is that a project like this at Hampshire would offer skills really important to learning how to be in charge of your own survival and moving towards a sustainable lifestyle. I know it's really important right now with climate change to reduce emissions and have the resources we need to survive regardless of what happens to grids and in governments. It's nice to be independent and self-reliant, and that's what I hope to do in my own life so I can provide for those around me and teach as well. It's incredibly important. Yeah. That's all.
Great. Thank you, Oscar.
Mhm.
Great. If people have questions, please put them in the chat or Q&A. And so I'll just start off by asking, so are you envisioning this would be something that could be built at Hampshire?
that was the initial idea, but I quickly confronted with the Kern building the difficulty of legal measures and permitting, and so more of the EARTHSHIP experience, trying to convey that and its importance.
So, do you think that this is something that you would recommend that Hampshire consider doing in the future?
Definitely. I think a lot of these technologies can be applied to retrofit infrastructure. So to make them more energy efficient and have less greenhouse emissions. And so that could be done as well for students to learn and future jobs and employment. I know that's a movement in the moment.
There's a question in the Q&A from Cindy about who you worked with on your committee.
Oh, yes. So I'd like to express a thank you to Chris Cianfrani, hydrology professor, as well as Tom Long, the assistant architecture and design professor.
And a follow-up about what you're doing next.
Yeah, so my family we're building an EARTHSHIP in White Salmon, Washington. So I'll get to build one start to finish, and hope to create it as an event so potentially Hampshire students could come out and earn credits and learn this as well.
Wow, that's fabulous.
Yeah, thank you.
That's very cool, Oscar. All right. Well, thank you. And John says, good luck with the construction. Oh, one more question here in the chat. Briefly, what kinds of things do you think might need to change in the way an EARTHSHIP is built to adapt to this local climate?
Yes. So EARTHSHIPs can be adapted to any climate. Originally, they're in Taos, where it's very dry, but they've been built in Canada, Alberta, Canada, where it gets extremely cold, like -30 degrees Fahrenheit, and yet the EARTHSHIP is very comfortable. There is a link to a video I can share. I don't know if I can find it right now, but they have built EARTHSHIPs in Canada that function very well, so very well adaptable.
Great, great. Thank you, Oscar.
Yeah.
And if you find it, you could put the link in the chat.
Yes, I will.
Okay, great. Okay, so we are on to our next speaker, and this will be Jess Roy. So Jess.
Hello. All right. All right, can everybody see that?
Yes.
So I did my Div 3 project on the formation of stress granules when affected by the neuropathology of Alzheimer's disease. And my committee members were John Castorino and Jane Caparis, who I owe so much to for dealing with me through this whole process. Oh, geez. All right, so as a little overview, so throughout Hampshire, I have been studying neuroscience, but I have really been focusing on Alzheimer's disease and specifically gene therapy for Alzheimer's disease. So then I decided to look into stress granules, which I will get in a little more in depth to what stress granules are, but they're basically our body's way of dealing with stress. And to see how Alzheimer's disease can affect stress granules, I transfected cells with tau and amyloid beta 42, which are two really main initiators, or what scientists right now think are main initiators of Alzheimer's disease, and then used some staining protocols to then determine if stress granules had been formed. So I'm going to start with overviewing Alzheimer's disease and overviewing stress granules before I kind of dive into exactly what I did for my project. But so Alzheimer's disease is the most common form of dementia, and it is currently the 6th leading cause of death in the US. And yet, even with such a high prevalence, we still don't know a whole lot about the disease. So Alzheimer's disease is a neurodegenerative illness, meaning that it is progressive and continues to get worse and worse the longer the patient has this illness. And it also causes a degradation of neuronal matter. So it kind of kills the neurons. It does a bunch of things that I'll also get into. But the main thing that this disease impacts is memory. So people with this disease, it's a dementia, so people with this disease tend to forget. And this Alzheimer's disease also has a heavy impact on mood regulation and other things like that. So there is no cure for this disease. And there are treatments available, but treatments for Alzheimer's disease are really complicated since it's pretty hard to diagnose the disease until the patient passes. Because you need to be able to see those like markers that I was talking about earlier, which is hard to do when the patient is still alive, unfortunately. So that makes treating the disease really hard. So kind of diving a little bit deeper into Alzheimer's disease, we have really three main things that are going to be affected in Alzheimer's disease. So first you have neurofibrillary tangles. These tangles are hyperphosphorylized tau that gets hyperphosphorylized to the point of falling off microtubules. So tau is what holds cells together. And when they become hyperphosphorylized, they're overproduced. And when they're overproduced, they fall off the microtubules and it causes the cell to completely disintegrate. The other thing we have is amyloid beta plaques. So these are amyloid peptides that get cleaved at a different site. So normally it's called the amyloid precursor protein gets cleaved at site 43, but in Alzheimer's disease, it gets cleaved at site 42 and it causes the ends of these peptides to be sticky. And then the third main change are epigenetic changes, which epigenetic changes affect the way our body processes our genetic code. So it doesn't necessarily change like your DNA or change anything like that, but it changes the way our body processes our DNA. So just kind of going a little bit deeper into all of that. Like I said, when the tau becomes hyperphosphorylized, it falls off the cell, and then that causes the cell to die. And all of those dead cells tangle up all together and cause noticeable tangles that you can see when you're looking under a microscope. And the same thing for plaques. You can see them when you look under a microscope, but they're the amyloid precursor protein, which we don't really know what this protein does outside of its impact in Alzheimer's disease, which also makes treatment a little bit difficult for that. But it gets cleaved at site 42, causing the protein to have sticky ends. And then those sticky ends pick up surrounding debris, surrounding neurofibrillary tangles, and cause these saucer-like structures. And then finally, for the epigenetic changes that happen, the main ones are DNA methylation and ribosomal and mitochondrial changes. So for DNA methylation is when a methyl group is added to DNA, and it usually inactivates the gene. And this process is heavily increased in Alzheimer's disease, which makes a ton of proteins turn off, or DNA turn off, or whatever that DNA translates into turn off. For the ribosome, so ribosomal biogenesis is the making of ribosomes, and we've seen that this is really heavily decreased in Alzheimer's disease, as well as mitochondrial DNA is significantly increased in Alzheimer's disease, which leads to cell death. So none of these things are very good. All of these are obviously really, really bad and lead to Alzheimer's disease, or so we think. Like I said, there are still a lot of question marks in Alzheimer's disease research. But moving on into stress granules. So stress granules are a type of biomolecular condensate. And what I mean when I say that is it's a non-membrane bound organelle in the cell. So these, like most organelles in our cells, they are held together by a membrane, and that's kind of how they stay. but biomolecular condensates are held together by liquid-liquid phase separation. And I'm not gonna really go into the details of that because that's even way over my head. Anybody who knows chemistry real well might be able to talk about that a little bit better. But so specifically, stress granules are non-membrane bound organelles that occur only after the cell is exposed to stress. So this stress can be disease, it can be temperature shock, it can be like adding chemicals to the cells. That's usually only when you're studying in lab would you get stress granules after like exposing the cell to chemicals. But recently, very recently, I had to change a few things in my div a few times through writing it because a new article would be published that went against something else that I had read before. So this very recently has been implicated in disease, which is really, really cool. So the assembly and disassembly of stress granules is a little bit of a complicated process, but basically it is caused by transitionally stalled mRNA binding to and then unattaching from protein interaction networks that consist of macromolecules with either 1, 2, or 3 interaction domains. So you can kind of see here, you have a cap, you have a bridge, and you have a node. And those all you can see here have those domains that the mRNA gets attached to. And that all attaching together causes this kind of little bubble of mRNA and proteins. And when this bubble occurs, it inactivates all of the mRNA and all of the proteins that are inside of this bubble. Right now, we've only been able to discover proteins that are mainly involved in like apoptosis and alternative splicing to be sequestered into stress granules. But like I said, research on this is so new that as I speak, a paper might be coming out completely disregarding all of that information. So it's a little hard to tell. But linking stress granules back into Alzheimer's disease. So the protein composition of stress granules, like I was kind of mentioning before, is mainly proteins and mRNA that's focused on apoptosis and alternative splicing. A lot of these proteins, which include TIA1, FUS, TDP43, and a bunch of others, they're all heavily implicated in different neurodegenerative diseases as well as cancer. So when these proteins are all sequestered into stress granules, they're rendered inactive. So in disease is when we see what's called stress granule hardening. So this means that when a stress granule is formed, it immediately hardens and will never be able to disassemble back into the cell even after the stress is removed. This is currently thought to be a pretty harmful process. But once I dive a little bit deeper into my results, I'm going to talk about why I don't believe that it is a harmful process, because it's holding on to these proteins that we currently think might cause Alzheimer's disease. So if it hardens and doesn't allow the proteins to interact with any of the neurons, that's probably a good thing.
You've got about a minute left.
Awesome. Okay, so I'm going to quickly go through my results. There are basically a bunch of pictures. So I looked at 3T3 cells, which are fibroblast cells, which aren't neuronal cells. Unfortunately, neuronal cells are really, really hard to work with. And as you can see here, my A through D is a non-stress cell, and my E through H is a stress cell. And you can see in the C and G, the red part, those are stress granules forming. So in G, you can see all of these tiny little dots. And this is after I exposed the cell to a chemical stress. So I exposed it to sodium arsenite for 30 minutes. And you can see here that that caused a lot of stress granules in the cell. So all of my variables that I use to test to see how Alzheimer's disease might affect stress granule formation is set up here. I'm not going to go through it. You guys can quickly read it, but basically I transfected cells with a hyperphosphorylized tau, the amyloid precursor protein, and then treated those cells with the cut version of APP, which is amyloid beta 42 that I was talking about that is sticky. So here we can see this is what happens when you just treat a cell with amyloid beta 42. You can see in this B panel over here a bunch of stress granules that have been formed. Here is the cells when they're just transfected with tau. So this green is going to be transfection and the red is always going to be what you want to look at to see if there's stress granules. So in this one, no stress granules were formed. We have here transfected tau as well as treated with amyloid beta 42. And if you look in this bottom panel, you can see the cells that express green don't have any stress granules, while the cells that aren't expressing any green look like they could have some stress granules. So that's very interesting. Here we have transfected tau treated with sodium arsenite just to show again that sodium arsenite causes these stress granules and that these cells can still get stress granules.
You're out of time, but you can go into your question and answer a little bit.
Okay, well, those were really, I just have a few other pictures here of transfected with APP. My results for APP were way less conclusive. It was harder to tell if these, if stress granules still formed when transfected with APP, but I did see that tau, the hyperphosphorylized tau, seemed to stop the production of stress granules, which I think means that stress granule hardening is protective, because then it stops these proteins from allowing to interact with tau and misfolding the cells and causing cell death. So yeah.
Okay, wow, thank you. That was a lot. That's amazing.
Yeah, sorry, there's a lot of information to throw in.
Very cool study. Spencer has a question. What are your future plans? Really interesting work.
So my future plans, my next, this summer I will, or after graduation, I'll be going to Massachusetts General Hospital to continue studying Alzheimer's disease in a lab there. So I'm hoping I can convince my PI to study some stress granules while I'm there. But other than that, I'm definitely trying to get my PhD eventually and continue studying stress granules.
Other questions you can put in the chat. We have another minute for any other question for Jess.
I see Todd asked, did any of your findings go against your previous assumptions or knowledge on the topic? Yes, absolutely. So I thought that tau and APP and A beta 42 were all going to cause stress granules. I definitely wasn't expecting tau to seem to prevent the formation of stress granules. But once I dove a little bit deeper into research of stress granules, I saw that there are some other things that prevent the formation of stress granules, like viruses and stuff. So it's really interesting.
Does anything from your research have words of wisdom for people with who are like elderly and facing the onsets of Alzheimer's?
I guess just that I think every day we're getting closer and closer to better treatments and kind of diving deeper into what it is that is causing Alzheimer's disease, because that is our biggest question right now of what's starting it and how can we stop that. Mm-hmm. We're getting there.
Great. Well, thank you so much, Jess. That was wonderful. Okay, we are on our next, for people joining us late, we are running about 5 or 6 minutes behind just because of the announcements at the beginning. So we are starting Sophie Kobar's study. So Sophie, take it away.
Hello guys. So my name is Sophie Kobar. I'm a Div 3 here with everybody else. I mostly focused on for the past year the SARS-CoV-2 pandemic. I'm an EMT firefighter. I'm also an ER tech, so an emergency room tech. And I've spent the last year really working in close with it. Let me put it in present mode, hold on there. My committee was Cindy Gill and Elizabeth Conlisk, and I'd always like to thank them for everything. They helped me a lot with this. It's been a heck of a year for all of us. I'm sure you guys can relate to that. So I'm going to go into it. So these are some pictures from the hospital where I was working. So my div covers 15 different aspects of the SARS-CoV-2 pandemic. I picked 3 sections to share with you guys today that I thought were the most important and pertinent. But my div, if you guys really want to read it, it's 15 sections. It covers all these topics listed here: past pandemics, viral structure, transmission, treatments, COVID and ambulances, healthcare burnout, masks and respirators, PPE and decon, testing for COVID, imaging, chest x-rays, ventilators, blood types, racial disparities, vaccines, and of course, emergency departments. Good half of it was probably narrative. I did have a few interviews with some of my fellow coworkers who are doctors, nurses, other frontline workers about working conditions and patient care and a lot of other stuff. So if you're more interested in that aspect of it, you can look at my div. So I'm going to go straight into it. So this is more about viral structure as one of the sections I'm going to tell you about today. So I don't know if you guys have seen cryo-EM data in the past, but cryo-EM is like a cryogenic image using electron microscope of usually an isolated particle. Usually it's cells. It has to be pretty small to be able to do it because we're using electron microscopes work. It has to be completely cryogenically froze all the way through. So it has to be pretty small. So here we have a viral particle of the SARS-CoV-2 itself. It's in color. It has a computer generated image over the actual electron microscope data here. So the yellow sections are viral RNA and their little vacuoles you can see there. That's what gets injected or put into our own cells. That's how it uses them. So we have a lipid bilayer here, much like our own. And then we got the protein spikes, the infamous protein spikes on the surface. SARS-CoV-2 is pretty cool. Most of the newer adaptations and mutations have come up with more invasive ways to evade the immune system. One of the ones I liked looking at was the viral protein spikes itself can actually mask itself using sugars. So they'll accumulate sugars over the spike so our immune system can't recognize the spike itself, because a lot of times that's how they're marked. So how it works, you have to come in contact with an active viral particle, usually through aerosol or a liquid droplet from like a cough or a sneeze. If someone's unmasked or they're like fairly close to unmasked, it can happen. Or if you're improperly wearing a mask, it can happen. I know many of us are vaccinated now, but it's still, you still got the chance there. So it gets into your respiratory system or a mucous membrane, and it tends to go to endothelial cells. So that's going to be like the lining, usually found in mucous membranes or any lining of the like inner surface of you. Generally the respiratory system though. It tends to prefer what are called ACE enzymes or angiotensin converting enzymes. There tends to be two types of angiotensin converting enzymes. The first type takes angiotensin 1 and converts it into angiotensin 2. And angiotensin is found in the renin and aldosterone system, which controls blood pressure in humans. But ACE2, they believe has some effect on cardiac contractions. And it seems to prefer, like the newer literature that's coming out is saying that it prefers ACE2 over ACE, the normal generic ACE. So the first one would affect blood pressure and the second one affects cardiac function. And the findings in COVID patients seem to align with that. A lot of COVID patients really suffer, obviously from respiratory first, but going into like blood pressure and things like that. And at late stage, a lot of cardiac issues occur. So it's really good to find out where it's coming from. So they do have vaccines now, which is fantastic. We're so blessed for that. We were waiting in the healthcare so long for these. So there's two general vaccines that I'm gonna tell you guys about. So the first is mRNA. You've heard about it before. This is the newer type of vaccine. It takes viral RNA and uses it and basically exposes your immune system to it without exposing you to a live virus. I say alive, but they have no metabolism, so they're not really alive, but active. And then there's a typical vaccine, typical I say, because it includes inactive viral parts. How long are they effective for? Originally, when they came out in December 2020, a lot of us were thinking, and the literature supported that they were at least 3 months. But compared to the little studies that are coming out, it seems that they're much longer. They're guessing 6 months and up for the effectiveness. I believe the CDC is planning about releasing information about boosters shortly. So we'll learn about that. It's likely to become like the flu vaccine every year. So transmission, social distancing, why? Yeah, so this one's a big, huge topic. So pretty much what they found is unmasked, and this has been data for a long time talking about flu and all the other things way back. The 15 to 35 feet respiratory particles can be projected through cough or sneeze. So it's really important to do masks. Even just a cloth mask prevents 50% of those particles from entering your respiratory system or oral pharynx, your mouth cavity. So there's 3 types of particles that can be projected from the respiratory system. Aerosolized, so that's any particles traveling through a gas. Liquid droplets, so that's any particles that are suspended in a liquid. And individual particles, they can be large or small. So we have a few different types of masks that I was going to show you that you guys probably already all know about, but you should know the data on them. So cloth masks just alone are 50%. That's not with a tight fit. That's not fixed appropriately. It's just slapping on your face. Surgical medical masks. So these are generally a polyester blend, sometimes made of different plastics that are statically charged. They're supposed to be mostly liquid proof. So they're going to stop mostly large particles in liquid droplets. They will not stop aerosolized. Same with cloth masks. When put together and doubled appropriately, they can almost be up to 90%. So they can almost compare to a medical grade respirator, which is what we were in the hospital. Medical grade respirators, though, of course, you need to be properly fitted. And there's a thing called a fit test where they actually put aerosolized particles within you in a contained space, and they test if you can like sense them and all that good stuff. So there's different grades of respirator. For surgical masks, there is such a variance on the statistics because most people do not appropriately fit them. 56% is if you just slap it on your face, you don't bend the metal, you don't try to like affix it to the shape of your face. What we're always going for is an airtight seal that will keep out 77% around the particles if it's like affixed to your face appropriately. Let's see. So back to vaccines, I forgot to tell you guys. So Moderna and Pfizer are mRNA vaccines, and Johnson & Johnson, the one that was having all the blood clot issues, is the old type of vaccine with the whole particle essentially in there. Mostly the risk was for individuals on birth control because that increases your chances of blood clotting. It's like 1 in a million chance. Yeah, so I believe that was my presentation. Questions, comments?
Great, thank you. That's really important work. Questions? Okay. Where are you going to take this work next? Or what are you going to do next?
I don't know. So I'm probably this summer, at least, I'm going to still be working part-time in the ER probably. And then medical school, hopefully. We'll see where it goes. But I love public health. I'm really interested in like virology and epidemiology. But yeah, 7 in the chat.
Yeah, the chat's lighting up with compliments. And anybody have any questions? When you were talking about doubling up masks, was that for the specifically the surgical masks or also cloth masks?
So the preferred or the official way to do it is you take a disposable surgical mask and then you fix it to your face the best you can using the metal tip and using the adjustable or turning the side, and then you put a cloth mask over it. And that was supposed to like prevent like too many usage of the disposable masks, because at the time production was not fantastically catching up with the need. So using your washable, your cloth mask over your like higher protection mask was supposedly supposed to save more of these, so you could reuse the cloth ones and wash them. But if that makes sense.
Cool. A question in the Q&A from Cindy. What was the hardest part of your Div 3?
I suppose reflecting on all the hospital work and cases and stuff. I like talking to people. I like informing people without being pushy, hopefully. Yeah, I think talking about past calls and stuff was the worst.
And Sarah Steely says in the chat, thank you so much. We couldn't have gotten through this year without you working on our testing and vaccine clinics. Thanks for putting your research and knowledge into action for the community safety. That's wonderful.
It was always fun. Thank you, Sarah.
Okay. Well, thanks very much. Oh, sorry, there's one more question. Oh, no. Okay, that one is done. All right, great. Thanks so much, Sophie.
Thank you, guys.
All right, wonderful. So our next presenter is Tucker Catanzaro. Tucker, take it away.
Hello, everyone. Thanks. Let me just share my screen really fast. Are you always able to see that? Okay, cool. Awesome. So for my div, recommendations for protecting recreational fisheries, examining differences between salmonid anglers and black bass anglers. So let's get into it. So just some quick background. So what is recreational fishing? So it's fishing for enjoyment and sport as opposed to commercial gain or sustenance. So it's enjoyed by millions across the United States. It's $1 billion industry each year, and participation ranges in it from doing it once at a summer camp, which I'm assuming some people in the Zoom call have done, to taking part in professional tournaments. Something else to note, most of the fish caught in recreational fishing are then released alive, as you see in the photo on the right, so they get to swim away and be a fish for another day. So what are the impacts and issues of recreational fisheries? So there's a misconception that recreational fisheries cause minimal damage. If you try to compare a commercial fishing boat, like a large trawler, to a single angler on the Swift River, they seem incomparable. How could one person contribute to harming a fish population? But if you take all the anglers on that river in one day, then all the anglers in that watershed, and then that state and so on, their impact becomes much greater and clearer and easier to compare. And so it's estimated that recreational fishing accounts for about 12% of the global fish harvest every year. So they do have an impact on the fish populations globally. An issue is that recreational data is not as readily available as commercial data. Individual anglers are not under any onus and responsibility to report their data to management agencies, and that data isn't collected as easily or as often. And so this lack of available data limits the management strategies that we can use. Traditionally, for commercial fisheries, we use statistical population models. But because we don't have as much data available for recreational fisheries, that's just not possible. And so how do we manage them? So instead of relying on models to check the population size and things like that, we instead rely on outreach, education, and behavior change as core parts of recreational fisheries management. So recreational fisheries management kind of walks this line between hard sciences and then also doing lots of outreach in social sciences. So more effort is spent changing the behaviors of the individual anglers that are out on the water. Unfortunately, not all anglers are engaging with fish conservation in the same way. We're able to get some anglers to handle their fish properly and some anglers to change their equipment, but we're not able to get them all interacting with fish conservation at the same level. And so black bass anglers, a species-specific group that I actually am a part of, are one of those groups that are lagging behind. And when we compare them to other groups, we find that they're taking slower to these ideas, to these conservation ideas. And the issue becomes, if we're leaving behind these anglers, then we're leaving behind fish. And so my question is, what are the factors contributing to the difference in how salmonid anglers and black bass anglers interact with fish conservation? So salmonidae, those are anglers trying to catch salmon and trout, like the brook trout you see above. And so they both have freshwater and anadromous populations. Anadromous mean they go from freshwater, they mature in saltwater, and then they come back to freshwater spawn. They have a lower thermal tolerance, meaning they prefer colder waters. They have a lower tolerance to low dissolved oxygen. And so because of this, they're more often found in cool, well-oxygenated bodies of water, such as streams, where the current helps to oxygenate the water where the water hits the rock, it'll help aerate the water below. Black bass, such as the largemouth bass above, reside entirely in freshwater. They have no need to migrate. Their populations are completely fine living in a 100-meter pond their entire life. They have a higher thermal tolerance, meaning they can stand warmer water temperatures in the summer. They have a higher tolerance to low dissolved oxygen, which often goes hand in hand with warmer water temperatures and also waters that have more nutrients in them. And they can be found in much warmer, stagnant waters because of this. Like I said previously, they're fine living in ponds their whole life. And studies show that they have lower rates of post-release mortality compared to trout. Being caught is a very, very stressful event for these fish. You know, you're catching a fish, it's being exercised throughout the water, and then you come and you take it out of the water and you expose it to air. So these are very physiologically stressful events. Blood cortisol, blood glucose, both increase with any fish that is being caught. But black bass have been shown in some studies to be able to withstand that event a bit better than salmonidae. And because of that, they have lower rates of post-release mortality. And so it's shown that black bass might be a little bit hardier. They might be able to withstand a little bit more handling. And even without knowing the life history, habitat preferences, physiological differences, just looking at these two fish, the black bass and the salmonidae, you can tell they look like pretty different fish, accustomed to different things. And so demographics. So fly anglers, fly fishing is the preferred method for catching salmonids. Fly anglers are more likely to be college educated and have higher incomes than other freshwater anglers. 45 of them had at least a bachelor's degree versus 32% of other anglers. 49% of them have incomes above 75,000 versus 40% of other anglers. And this doesn't necessarily impact an ability to understand scientific outreach. It's been shown that experienced anglers, regardless of their background in education, have the same level of comprehension to conservation ideas. But this illustrates how the two disciplines have very different audiences and how one outreach method might not work for all of them. And so non-governmental organizations. So the unfortunate fact is that government agencies are unable to do all the necessary outreach to protect these fisheries. There's simply not enough time and labor to cover every pond, river, lake, and coastal area where people are fishing for fun. Because of this, we've had NGOs that have stepped up to help fill this gap and protect certain species, bodies of water, and geographic areas. I've looked at a lot of them. The two I'm going to talk about really fast are BASS, the Bass Angler Sportsman Society, and Trout Unlimited. BASS is the main NGO for black bass fishing. They focus on habitat improvement, supplementing government projects, stocking fish, and other conservation efforts. And they do so through a nationwide network of local chapters. Trout Unlimited focuses on cold water fisheries, mainly salmon and trout. They share the same goals of habitat protection, outreach, and advocacy, and they also do so through a large nationwide chapter. So the differences are that Trout Unlimited is a nonprofit conservation, and conservation is their main goal. BASS is for-profit. They're part of a larger media company, and the conservation arm is only one part of their larger organization and goal. Most of their time is spent maintaining competitive fishing tournaments. And so because of that, the conservation goals kind of get pushed to the side of everything else. So Trout Unlimited seems to advocate for goals that lead to long-term conservation, and BASS seems to advocate for goals that improve short-term recreation. And kind of just reinforcing this, looking at their social media accounts on YouTube and Instagram, I looked at a calendar year of posts, and in the 1,200 posts that BASS made, 0 of them were about conservation. Trout Unlimited at least had some, but it's obvious that BASS, at least in outreach on social media, has some other goals than conservation. And looking at tournament angling, like I previously mentioned, these are the tournaments that BASS is trying to maintain commercial success in. So it's all about catching fish competitively. They are popular in both disciplines, but they're much more popular in black bass fishing, with over 22,000 annual tournaments, ranging from a bunch of friends getting together one day to just go catch some fish to multi-million-dollar tournaments that rival a large sporting event. The issues is that these tournaments can exhibit harmful fish handling, like the photo you see on the right. These fish are being air exposed for an unnecessarily long amount of time. And this compounds the effects of the fish's physiological response, from being taken out of water the first time, put in a live well, put in a tank on a boat, and then taken out again and put in the tank, taken out again. Every time you do that, it compounds the fish's physiological response and the effects that they're feeling. And so steps are taken to reduce fish mortality, but they're not always effective. There are always fish in these tournaments that don't return to where they were caught and will die after, unfortunately. And so these tournaments just kind of exhibit poor fish handling and fish conservation at times. And so just kind of a summary of the factors I've been identifying through this. So the species have very different life histories and habitat preferences, whether that be the type of water body they're in, the range they're in the United States, or where they are during certain seasons. Anglers in the two disciplines vary greatly demographically in terms of income and also education, meaning that we might want to cater our outreach methods differently. Salmonid anglers seem to focus more on long-term conservation rather than short-term recreation goals. And no nationwide nonprofit exists to advocate for black bass conservation. There's a huge gap in the sport. The popularity of bass tournaments help to reinforce poor handling, and black bass anglers appear less likely to see conservation talked about online. So my recommendations. So to create behavior change in the sport, we need to create social norms. We need to make people feel like it's expected of them to treat fish in a responsible and conservation-minded way. So to do that, sorry,
Yeah.
sorry, I heard something.
I was just saying just about a minute.
Oh, thank you. So one of my recommendations is to create a nonprofit that works to conserve black bass, something similar to Trout Unlimited, that won't have its goals affected by being part of a larger media conglomerate. And we need to make black bass anglers more aware of the species-specific issues that they face, create education materials that focus solely on bass, and work with tournament organizations to change their formats. There are alternative formats that allow for competitive fishing to stay alive, but also to help improve the outcome of the fish that are caught. In conclusion, we're trying to balance wanting to exploit the resource with needing to conserve it. Some on-it anglers have struck this balance earlier and more effectively, but by using existing avenues for outreach and creating new ones, we can begin to reach out specifically to black bass anglers. It's becoming obvious that the two disciplines each require separate, unique methods of outreach as they differ greatly. So just takeaway, to best protect this natural resource, we need to work to educate all the anglers. And I just have some long list of references, and that's it.
Wonderful. Thank you, Tucker.
Yep.
That's really interesting work. And we have a question in the chat, in the Q&A from Spencer. A very informative presentation. How do you think NGOs can better advance their conservation strategies or goals using social media?
So I think part of it is that these NGOs just haven't really had to change their ways in so long. There are smaller NGOs we've seen recently that have used social media in really effective ways to reach out directly to anglers that are following them. So I think just having, maybe like a weekly tip saying this is a good fish landing to, Fish Friday, maybe something like that, to at least dedicate a distinct portion of their social media presence to conservation, to make it so it's a regular thing, would make a big difference. As it stands now, for groups like BASS, it's something that either happens never or doesn't happen on a regular basis. Yeah. Mm-hmm.
And we have a question from Sarah Steely in the chat. What kind of mortality rates are there in non-competitive catch and release fly fishing?
I'm not entirely sure. The post-race mortality sometimes can be hard to monitor. It can be things like it's a smaller fish. It can be things like a fish is taken off a nest and its brood is eaten by a smaller fish that comes in during that moment. It can be things like a fish is released and it simply can't predate as effectively and it dies a few weeks later. It can be things like a fish is released and then a larger fish comes along and eats it since it can't swim as effectively during that time period. So unfortunately, I don't know the exact rates, but it's something where there are a lot of different factors that can lead to post-release mortality. So it's not a very cut and dry kind of thing.
Mm-hmm.
Mm-hmm.
Great, thank you.
Mm-hmm.
And there's lots of great comments in the chat. It's more complicated than I thought.
It's definitely a hard thing to keep track of sometimes.
Yeah. What are your next plans? Are you going to follow up on this work, or what are you doing next?
Yeah, I would love to follow up on it. It's something I'm very passionate about. I would love to one day work in a state or federal agency doing angler outreach and education. I've been looking at job postings, and it's encouraging to see more state agencies that are posting jobs that specifically have to deal with recreational fisheries management. So it's encouraging to see, and hopefully I can take one of those jobs one day.
Great, wonderful.
Awesome.
All right, I think that's it. Thank you, Tucker.
Yeah.
Okay, we have in this session, current session, we have one more presentation, and then we'll be taking a short break. So Molly Dent, take it away. Hi, Molly.
Hi. Alrighty, share my screen. Here we go. Okay. Awesome. So I'm Molly. I'm a Div 3, and my Div 3 is focused on conservation and bioacoustics, specifically developing a stranding alert network to prevent fatal mass strandings of small cetaceans near Wellfleet, Massachusetts. Basically, that just means we were looking at whether or not we could predict mass strandings of dolphins using their acoustics and vocalizations. So what is a mass stranding event? A mass stranding event is defined as at least 2 cetaceans stranding at the same time in the same geographical place. And it's been super heavily debated about why these occur, but no one's really been able to come up with a consensus as to why they're happening. And because we don't know why they're happening, we obviously cannot predict them in order to prevent them. Here is just a map of Wellfleet Harbor, which was our study area. It's off of Cape Cod, and these bigger circles just mean that more animals stranded here. So it's pretty clear that Wellfleet Harbor and the area surrounding it are hotspots for strandings. And basically, ever since strandings have been recorded, Wellfleet Harbor and the areas around it have been super heavily affected by strandings. So the point of an early warning system would be to cut down the time that it takes for stranding networks to respond to these strandings. The variables that determine likelihood of these animals surviving is time spent on land, so how long they're out of the water, thermal stress, whether that's hot in the summer or cold in the winter, and separation from conspecifics, because they're very social animals, so when they're separated, it stresses them out a ton. And so the faster these stranding alert networks can get to the site, the better. A lot of times they're able to just like herd the animals offshore before they even strand, and that just minimizes a ton of these super negative stressors. The reason that we chose acoustic behavior to try to predict these events is because these acoustic cues are really like specific, and there are a lot of variables that can be like differentiated between. So a signature whistle specific to dolphins is unique to every animal. So because dolphins live in the ocean, they rely really heavily on sound to communicate with each other. And signature whistles are like unique to each dolphin. It's basically like a name, and they use them for individual identification. And studies showed that when stressed, bottlenose dolphins specifically were shown to increase production of signature whistles. And so because strandings are so stressful, we figured that this might apply in this situation to other dolphin species. So we did specifically look at dolphins. There are other animals that strand, like pilot whales and whales, but because dolphins whistle and we'd be able to learn more about their vocalizations through those whistles, we looked at dolphins. So we looked at the short-beaked common dolphin and the Atlantic white-sided dolphin, which are 2 species that strand extremely all the time in Wellfleet Harbor. So for our methods, I actually, despite COVID, got to do some data collection, which was really exciting. There's me on the boat with my little sound recorder there. So every 3 weeks or so, we'd go out to Wellfleet Harbor, which is right here, and we put a little buoy with a sound recorder right where the star is. And we made sure to switch it out every 3 weeks or so, just to make sure that it was always recording and that the data hadn't like filled up. So for acoustic analysis, we used Raven Pro version 1.5. It maps frequency and time on these axes, and so that you can like look at whistles visually in order to like analyze them. This is a little bout of a signature whistle right here. You can see that it just like it's repeated and it looks pretty much the same in all of these instances. So there was 12 mass stranding events that we looked at, and we decided to pick 24 corresponding non-stranding days, like within 2 months of those strandings, so we could kind of pair them environmentally. And we used the SIG ID method for free swimming bottlenose dolphins, which again helps us classify these signature whistles. So for statistical analysis, we also wanted to look at just like general presence of whistles to see if like we could just predict a stranding based on like whether or not there were whistles picked up by a recorder. It was significant, which is exciting, but the way we did this was looking at whether or not there were detections or no detections, and then whether or not there was a stranding or not a stranding. And then for acoustic cues, we looked at signature whistles, and we also looked at intersignature whistle intervals, like time between whistles, and overall whistle rates, and compared those between non-stranding and pre-stranding days. One thing, just to kind of increase our accuracy of a stranding alert network, was we decided to add environmental parameters kind of into our mix. We looked at water temperature, time of year, season, wind speed and velocity, and lunar phase, just to see whether or not like we could figure out when these strandings were happening locally. Obviously, no one's really been able to like figure out why like all dolphins strand, but we figured because it's like in a smaller area, we might be able to find some trends because we have like very long-term data. So acoustic results, we looked at a lot of whistles, 7,711 whistles, and we found 2,637 call types, 998 of which were signature whistles. So before, we had been looking at 48 hours before a stranding and determining that as pre-stranding, but now we've decided to look at just 12 hours before a stranding to kind of minimize that time. So we found in preliminary results, we haven't like tested for significance, but 82% of signature whistles in recordings within 12 hours versus 54% signature whistles in non-stranding recordings. So this this is similar to what we predicted. And if these are significant, and if like we can go on to maybe find more of these trends in other recordings, then there might be a pretty solid basis for an acoustic alert system. Our detection, just like general acoustic detection, so like whether or not there were whistles and whether or not there were strandings, that was also significant with a p-value of less than 0.01. So that was also exciting. So these are just two acoustic things to kind of base an alert system on. Environmental results were also exciting. Using a Welch's t-test, we got a p-value of 0.005 in terms of higher wind speeds immediately before stranding. And 56% of our strandings occurred like when water temperature was below 0.6 degrees. So there was 3 months during which this happened, and over half of the strandings occurred then, which was exciting to see. So in terms of conclusions, we found that dolphin whistles were more likely to be detected on pre-stranding days just in general, and preliminary findings suggested that higher percentages of signature whistles occurred in the 12 hours before a stranding. Again, it was 82% versus 54%. And so using these as a basis, but also combining them with environmental variables like wind speed and water temperature, we're hoping to create the most accurate alert system possible. So on any given day, we would hopefully be able to like give a percentage of likelihood of a stranding. And then IFOC can get out there as fast as possible. And again, hopefully just prevent the dolphins from getting on shore entirely. This is a pretty big project. I hopefully will be doing this after I graduate. Next steps include, again, decreasing the pre-stranding time frame from 48 hours to 12 hours, which we're already working on. We also hope to deploy recorders in different places in Wellfleet, and maybe we'll get funding for another recorder so that we can kind of just be more likely to capture whistles as they're happening. We plan to extend our time frame for environmental parameters. So we were only looking at a time period of 3 years, but hopefully we can do like 20 years. And ideally, I will be publishing a paper on just general acoustic behavior of these two species and attending the Society of Marine Mammals conference in December, talking about this. My references and my acknowledgements, because I have a lot of people to thank. Awesome. Yeah, I can definitely take any questions.
Wonderful. Thank you, Molly. Really interesting work. So please put your questions in the chat or the Q&A. And I'm curious about the wind speed. You mentioned that wind speed was higher. Is that how do you think that? Do you think that's causal to the strandings, or how do you think that's related to the like why do they strand when is there a reason they would strand with high wind?
I'm not really sure. I think that Layla, my advisor, came up with that as a thing to look at. A lot of the times, like the dolphins are kind of just getting stuck in like the mud area that is part of Wellfleet Harbor, and some people think it's just like the weird like oceanography of that little area. It's kind of like suddenly shallow and like tides are weird, but yeah, we're not really sure. I don't know if it's causal, but it's just an interesting like environmental parameter. I think it'd be really cool to look more into that, to like see, because other papers have suggested wind speed, but like I don't remember their reasoning for looking at that. So yeah, definitely something to keep in mind.
Great. And Oscar asks about listening to the dolphin recordings, if you can explain more about how you can listen to them.
Yeah, it's a really good question. I can probably like put up somewhere some examples. There's like a ton of, if you just like search like bottlenose dolphin acoustics, I'm sure that you can find some like really cool examples of whistles, because they're very, very heavily studied. And these two species have not been studied. Actually, just finding the fact that they produce signature whistles is like really exciting research in itself. And that's what I'm going to be presenting on at the conference this December. But yeah, we've got like just like a big database of all of these recordings basically. And I download them and like listen to them. And like if you find a recording like online, you should definitely try to listen to it at a slower speed, so like 0.25 speed, and that way you can really like hear the kind of contour of the whistle. It's really neat.
So we have time for another question if somebody has another one, and I'll just ask in the meantime. So would you expect then that you could use this as a predictive framework by having like hydrophones in the water and listening for increases in proportion of signature whistles in certain populations?
Yeah. So it's something that's kind of like happening long term, and that like wouldn't be a super short term thing. But I know Layla has been working with Google to try to like create a like, what's the word? Like a remote and like kind of AI basically that would like be able to like real time analyze these recordings in order to like just immediately be able to like figure out when these increased proportions of signature whistles are happening. So that's been really, really interesting to hear about. It's just like working with like this really big company and trying to figure out if a computer can do all the work for us, just in a very, very speedy time frame.
Interesting, great. Two more questions have come in. One from Flynn in the Q&A, wondering if other folks are doing any similar research with predicting strandings, and if so, have you been able to connect and talk with them?
I don't think so. People have been like pretty interested in the environmental parameters, but I haven't really heard of people who are trying to predict strandings like acoustically. And I've talked a lot with IFA, the International Fund for Animal Welfare, who's provided us with all the data. And they kind of have like their own like ideas. A lot of it's just like, they think that it's like the mud and like the weird like way that the ocean bottom increases. But yeah, it's pretty like new, I think. I don't think a lot of people, I mean, Layla's been doing this project for a while, but.
Great, so you're on the cutting edge. That's wonderful.
Yes.
And Dula asks, what are the frequencies of the calls? Do they vary with the species?
Yeah, I think most dolphins, oh my gosh, I should know this. I think it's definitely kilohertz. Usually like between, I think like 4 and 24 kilohertz for dolphins. And like whales are like way, way lower. You have to like speed them up to listen to them. But yeah, between those ranges, and dolphins can hear like a super, super wide range of frequencies, unlike people. We can't hear a lot of dolphin whistles unless we slow them down.
Great. Great. Well, thank you, Molly. Layla adds about wind speed. We're looking into different factors that could increase chances of animals getting pushed in towards shore. That's really cool. Very interesting work. Thank you very much. And I want to extend a great thank you to all of the Div 3 students who presented in this session so far. Really fabulous work. Yes, cheers, clap, clap. And we're going to be taking a short break, about a 10-12 minute break here, and then we'll be starting up the next half of the Environments and Change session at 3 o'clock, and Dula will be moderating that. So thanks very much, and please join us at 3 o'clock after a short break. I forgot to say thanks to the audience as well for asking all the wonderful questions.